So thank you, Bill. And I, of course, want to thank all of you for attending this event, giving me an opportunity to tell you about some of the work that's going on in my laboratory here at the Salk, and especially for your support of the institution. So if I can get this to work, what I thought I would tell you today, talk to you about today, is this cellular structure called the cilium, or there's more of them, they're called cilia. And the reason I wanted to tell you about this is over the last 10 years, this structure has basically been reinvented. It's gone through this transformation, and it's due in part to a collaboration between human geneticists who are interested in diseases that cause particularly congenital defects or inherited defects that have developmental consequences, in combination with cell biologists or people who work on cells. And what this has done is really transformed how we think about these structures and how they contribute to human biology and human disease. So I'm going to start by telling you what is a cilia, what they are, and then I'll spend some time talking about the work that's been done in human genetics that has sort of revealed their new functions. And then I'll end on a short story that has recently come out of my lab, which has really cast some light on how this structure, this cilium, comes about in the lung. So what are cilia? Well, they're these hair-like appendages that stick out of cells, and an example of that shown here. In this case, you're looking down on a cell that's not extending one of these, but hundreds of them. And so this is called a multiciliated cell. Most cells would only extend one of these. And these cilia are motile. They're beating away, and they're moving fluid across the surface of this tissue. Most cilia, as I'll get into, are actually immotile and act as little cellular antennas. Now, the origins of this structure actually go way back, millions of years ago, and they were thought to have arisen on unicellular organisms that needed a way of getting through the pond. And so we still see that today. So this is an example of a blue-green algae, known to you as pond scum, which is actually a plant that photosynthesizes, so it wants to be near light. And so the way it does that is to extend these two long appendages, which are also called flagella, but they're actually cilia. And as you can see in this movie here, it allows the organism to do a breaststroke through the pond to find its way to the best source of light. Now, during evolution, that unicellular organism became multicellular, but it retained this ability to make this cellular structure. And now you find them all over the organism, and now they begin to do different things. So it's not only involved in motility, but in this case, this marine organism, they're beating away and they're helping to move food into the digestive tract. And then as evolution proceeded further, as we got plants, particularly land-based plants, this structure was lost. They decided they don't need it, and they've lost even most of the genes that are required to form it. But in animals, they diversified even further, and now you find them ubiquitous on all parts of our tissues and most of our cells, and now they carry out very different functions. And two of the functions are illustrated in this scanning electron micrograph shown here. This little guy here is the sperm, and like blue-green algae, it's a single cell which extends this long motile cilium out the back, which is called the flagella or the tail. And of course, that's motile, and it allows the sperm to find its way up to fertilize its target, the female gamete. Now, that sperm is lying inside this structure called the fallopian tubes, which is the lumen where fertilization takes place. And the cells in the lumen extend hundreds of cilia, shown here, up into the lumen, and they beat like crazy, and they move the fertilized egg along the fallopian tube to the uterus. And so this slide, I think, nicely illustrates two of the long-known uses of cilia in human biology, which is in reproduction, and that it's required both for the male and the female in order for reproduction to work. Now, this turns out to be just the tip of the iceberg for these guys, because if you look at most cells in the body, except for blood cells, every single cell still extends a cilium. It's non-motile. It's lost the ability to move. It's a very short, stumpy structure, pretty hard to see. And this is two examples. One is on a neuron. So the neuron's here. It's got all these processes. It's extending this little short cilium. And this is a glial cell extending these little short cilium. For many years, people looked at these and they said, well, they're so small, they're vestigial in nature. Cells can make them, but there's no reason to have them. They're no longer important. And this was true in textbooks up until about 15 years ago. Now, the view of these structures began to change dramatically in the 90s when human geneticists began to work on an inherited disease called polycystic kidney disease. It's a very common form of end-stage kidney failure. It's pretty common. It affects about 0.1 to 0.25% of the population. And the way it works is if you inherit one mutant allele of a gene, then something happens over time in the kidney, and a kidney cell will lose the other functional copy. And now you have no functional copies, and the cell responds to that by beginning to grow. It doesn't become a tumor, but it proliferates abnormally, and this leads to the formation of these cysts, which eventually fill up, see if I can get this to work, fill up with liquid, and that affects the functioning of the kidney. Now, because it's a fairly common disease, human geneticists were pretty successful at mapping the genes that were involved in this disease and cloning them, and that led to the identification of two genes. One's called PKD1, the other one 2, that account for a large fraction of the people who come down with this disease. And in November of 2000, that's led to this Time article, because for the first time, you could genetically test individuals and figure out whether they were at risk at getting this disease or not. Now, that was a very important step forward, but we still had no idea, well, what does PKD1 and PKD2 do for a kidney? Why is it so important in order to maintain its normal morphology? And the field languished for a while until a very important observation was made. And the observation is shown on this slide. If you make a section through a kidney, as shown here, this is one of the tubules where the liquid flows and where the filtration takes place. And these kidney cells extend these little tiny short stumpy cilia into the lumen of this tubule. And it turns out that the products of those two genes, PKD1 and PKD2, localize to the primary cilium. And when that was realized, that set off a whole host of experiments to figure out, well, why is this little structure important for maintaining tissue homeostasis in the kidney? And the model that's emerged over the next 10 years is illustrated on this slide. What happens is that the cilium extends out of the kidney cell. PKD1 and 2 are in the membrane. When flow comes along, produced by movement of liquid through the tubules, it bends the cilium over, and that sends a signal inside the cell that says, everything's fine, don't do anything. But if these two proteins are lost, that signal no longer goes into the cell. The cell thinks something's wrong, begins to proliferate, and it eventually leads to these large cysts that cause the disease. Now, the second realization was that if you not only lost these proteins within the primary cilium, if you lose the cilium itself, you get the same disease. And that quickly led to the discovery of a whole host of different disease syndromes, which are shown here on the left. Genes that are associated with those syndromes shown here in the middle, and then finally the pathologies that are associated with each of these genetically inherited diseases. And what you can see is the common pathology is kidney cysts. That typically arises, but then there are pathologies in other tissues. And then subsequent analysis of all these different genes have shown that they all localize to the cilium, and they have something to do with either the ability to form the cilium or the ability for the cilium to function. And as you can see, when that happens, you get this wide spectrum of pathologies in different organs. So you get liver fibrosis, you get retinal degeneration, you get loss of smell, you get male infertility, you get obesity, you get brain malformations, all of which hint to the idea that if the primary cilium is not working in different parts of the body, then something bad is going to happen. And so where the field is currently at is trying to understand what the function or what the nature of the importance is for these primary cilia that you find on different types of cells, such as neurons and glia, and how these might be necessary for the normal function of these particular tissues where these cells reside. Now, at the same time, while there's sort of a reappreciation of what these little sensory cilia are doing, there's also been a dramatic change in what we think motile cilia are doing. So the place you find most of the motile cilia is in the lungs. So the lung is basically an airway. So there's your trachea. You come down to the proximal, the bronchi, into these branching and branching structures until you get to the very end where the gas exchange is going to occur. Now, every time you take a breath, you bring in pathogens, irritants, nasty things. And so the lung had to find a way of getting all of this stuff back out. And so what it came up with is this on the, shown here on the right, which are these cells which basically line all the airways from the top to the bottom of the lung, which extend these multiple cilia, as shown here, and they all beat in unison and basically move all that stuff back out of your lung to keep the airways clear. Now, the scale here is, I think, pretty important. Each one of these cilium it's only about 1/100th of an inch. They beat like crazy, and yet it's what they're capable of doing by beating in unison and all in the same direction is they're able to move stuff out of your lungs at a rate of about 10 feet per hour. So every couple times per hour, you're basically clearing everything out of your airways in order to keep the lung healthy. Now, as you can imagine, if this process stops, something bad is going to happen. And indeed, there's a number of diseases that are associated with ciliary dyskinesia, which is another way of saying that the cilia just aren't functioning properly. One form are the acquired diseases, which are not due to defects in the multiciliated cells themselves, but are due to damage to the multiciliated cells. Since they're out in the open there and they're constantly being exposed to stuff, it's easy to damage them. And smoking is a classic example of this because of the chemicals present in smoke. Cystic fibrosis is another example. This is an inherited disease that also causes the mucus, which is secreted by other cells in the lung and which have to be cleared out along with all the debris and whatnot. This eventually, because the cystic fibrosis basically causes the mucus to become more viscous, the multiciliated cells just can't move it out, and they eventually kill themselves trying to. And then finally, there's asthma, where you have these recurring inflammations of the lung, which basically damage the multiciliated cells after each episode. And this is a sort of a negative feedback loop. So if the multiciliated cells become damaged, then they're less effective at moving all these irritants and pathogens out, which lead to more damage, more inflammation, and that this keeps spiraling down until eventually the lungs clog up with mucus and there's morbidity. Now, in addition to these diseases, there's also something called primary ciliary dyskinesia, which is a disease that arises when you have an inherited mutation that affects the formation of the ciliated cell itself. And when these patients appear in the clinic, it's inevitably at birth because they have a severe respiratory distress, because they can't clear things out of the lung. Later in life, they're, of course, sterile for the reasons I alluded to in the slide that I showed to you earlier. And they also have water on the brain or hydrocephaly, which is due to the fact that the central canal of the brain is coated with multiciliated cells, and this is the way you propel the cerebral spinal fluid through the ventricles. Now, these patients, which have been studied for 50 years, have also come into the clinic, and about 50% of them have what's called situs inversus. So what is situs inversus? So if you look at down my midline, I'm pretty symmetrical on my left and right side. But if you go internally into the organs, they have a sidedness. Your spleen's on your left side, and your liver's on your right side. Your heart, the inflow and outflow tracts are oriented in a certain relationship to that left-right axis. And half the people who have primary ciliary dyskinesia or immotile cilia have this axis completely reversed 180 degrees. So this was known for 50 years, but it wasn't clear to anybody why a loss of motile cilia would lead to such a profound impact on the orientation of organs in the body. And it wasn't until a Japanese scientist, Hamada, who was very prospective, about 15 years ago now, realized that there are motile cilia present in the very early embryo. And so if you go back to the early embryo, this is, I think, a rabbit embryo, but the human would look about the same. At this stage, it's pretty much sheets of cells. There are no organs that have formed yet. But if you go back to the very end of the embryo, there's this little structure called Henson's node, which is shown here in the scanning EM. And if you look more closely in that region, what you see is that all the cells are sprouting cilia. Now, that's not too surprising because I told you cilia are present on most cells in the human body. But these cilia in the middle, I don't know if you can see this in the movie, they're actually all beating away in this circular motion. And what Hamada realized is that beating actually causes a directed flow across the embryo that goes from right to left. In other words, you get a flow that moves across the embryo from one side to the other. And so that then led to a whole host of experiments that have led to the model shown here, which I think is pretty well accepted now, which is that in the Henson's node, very early in the embryo, if we take a cross-section, there are two types of ciliated cells. There are cells in the middle, which extend a single cell, a cilium, which beats and generates this so-called nodal flow that moves right to left. And then there, around that, those motile cilia are sensory cilia, ciliated cells, which respond to that flow, much like the cilia in the kidney that I told you about earlier. And so the ones on the left get bent over in response to that flow. They send a signal into the cell, and this basically tells these cells on this side that you're going to be the left side of the embryo. And the cells on the opposite side become the right side by default. And you can nicely test this by making animals where they don't have motile cilia, and then you can blow on this in either direction and basically set which side is going to be the left and right side within the early embryo. Now, the reason this becomes very important is, as I told you, in the case of situs inversus, there's not much wrong with these people. You can reverse everything 180 degrees, and that's all right. What's bad is if it becomes ambiguous. If both sides think they're left sides, both sides think they're right sides, some cells on the left side think they're left, some think they're right, because inevitably what happens is that the organs basically don't know quite where they are and how to connect up with each other. And this is particularly true in the heart. And indeed, one of the major causes of congenital heart defects that people have been working on recently are thought to maybe be due to defects in this ciliary process that initially set up the left-right axis back in the early embryo. And so there's a lot of work now, particularly in medical schools, where they're going in and sequencing people who have congenital heart defects and then trying to identify the gene, and much of that work then revolves back to trying to figure out how those genes are involved in this early left-right patterning process that occurs in the early embryo. Now I want to end by telling you something about a recent finding in our laboratory, and this has to do with a special form of primary ciliary dyskinesia, where you have the patients are born with all these symptoms, severe respiratory distress later on in life, they're sterile, water on the brain, but unlike other forms of PCD, these patients don't just have non-moving cilia, they just don't have any cilia at all. And so this is called a congenital mucociliary clearance disorder with reduced generation of multiple motile cilia, which is also abbreviated as RGMC, and I'll try to stick with that. So this is what these patients look like. Instead of the normal pattern of multiciliated cells in the lung, basically nothing there. There's a stumpy cilia, primary cilia somewhere. The problem is that this disease is pretty rare, and so the families aren't big enough to map the gene to see what's causing this particular defect. They know it's somewhere on chromosome 5, and they think it's somewhere down here near Q11, but it's in too big of a region to figure out what genes might be affected. Now, we took this problem on 5 years ago, and we were looking for an experimental model where we could study multiciliated cells. And the model we came up with is shown here, which is the frog larval skin. And you might ask me, well, why is that a particularly good model? Well, frogs develop outside the female, so within 24 hours after fertilization, we have this little embryo shown here. And it turns out that the skin in frogs, because you're living out in the extracellular environment, they're sort of like the lungs and the kidney of the early embryo. The internal organs haven't developed yet. And so it's respiring through its lungs, it's pumping ions, it's respiring through the skin like the lung does, it's pumping ions in and out of the embryo. And so basically the skin has to differentiate into various cell types very early on in order to achieve these physiological functions. And one of the things it does, each one of these brown dots is a multiciliated cell that form across the embryo. So the advantage of this is it's on the outside, which makes it very easy to image. And here's an example of where we're imaging these multiciliated cells in the frog skin. There's a red dye here which shows you where the cells are. So these large cells here are actually mucus-secreting cells, just like in the lung. There's some other smaller cells that are involved in ion transport. You can stain them with blue, which picks up these cilia. So you can see these are the multiciliated cells. And because it's very hard to count cilia, it's like counting spaghetti in a bowl of spaghetti, we also put in a marker for the structure at the base of the cilium that nucleates its outgrowth. It's called the basal body. But each one of these green dots represents a basal body that's docked at the membrane as an extending a cilia. So we can go in and count these, and it makes me sick to look at this because I don't know how many of these things I've counted, but this is about 150 to 200 cilia that form in each one of these cells. So this gave us a nice assay to study these cells. We then began to look at genes that might be involved in making these cells, and we came across this gene we called multicillin. And the reason for that is shown in these two experiments here. So here's the normal pattern of cells. This is an embryo where we took multicillin away, and you can see the ciliated cells have completely disappeared. It looks like these patients that have RGMC. The more informative experiment to us is this experiment over here where we took multicillin, which is normally only present in these cells. It's only expressed in these cells, and expressed it in every other cell within the skin. And what you can see is that it alone was sufficient to convert all the skin cells into multiciliated cells, as shown here. Now, there's a lot of other detail here, but when this paper came out, the human geneticists that were working on this disease said, well, where is multicillin located in the human genome? And it turns out it's located on chromosome 5 in this region basically close to where these mutations that cause RGMC mapped. And indeed, this whole region of the human genome is highly conserved all the way down the frog. And it contains a series of 3 genes which were completely unstudied in any system. Nobody knew anything about these 3 genes. So about 2 months ago, we got a call from the human geneticists who said, well, we've now gone in and sequenced these genes in the patients that have this disease, RGMC, and the first 4 families they looked at had 8 individuals, all of which have mutations in multicillin. They said the next 20 individuals we looked at or so have mutations in this gene right next to it called cyclin O, and so that led us to go back and ask, well, cyclin O also required for the formation of these cells, and indeed it is in frog. So very quickly we've gone from knowing about some genes in frog to basically defining a locus on the human chromosome 5, which seems to be instrumental in just doing this one thing, which is to generate these multiciliated cells. So what we're trying to do now is to figure out how multicillin and cyclin O do this. How do they make a cell grow hundreds of cilia? We're also collaborating with Inder Verma's lab, a postdoc of his, Amy Firth, who has pioneered the use of iPS cells to study human lung disease. So as you know, you can take skin cells from these patients, put them into tissue culture, convert them back to embryonic stem cells by using the right cocktail of genes. They become pluripotent again, and then you can drive them, if you have the right tissue culture conditions, back to lung cells to see what happens. So we're now getting patients, the cells from the patients from our collaborators in Germany to basically generate these iPS cells at the Salk Institute so that we can formally test our model. What we need to do is to take our genes and put them back into these cells and show that they recover the ability to make multiciliated cells. And this will also pioneer the use of gene therapy to correct these gene defects. So I wanted to emphasize 3 take-home messages. One is you need cilia. When you go home tonight, thank the cilia in your lung because they've been beating like crazy all day long to keep your airways free of infection. I hope to have illustrated the sort of the combination, the power of human genetics with laboratory experimental systems. I hope this becomes a theme at the Salk Institute, and we find more faculty members who combine human genetics and model systems to gain insight into human biology. And then the last point is a truism, I think, among scientists, but I hope it's also apparent to the outside world, which is that even after the sequencing of the human genome, where we know all the genes, most of the genes, we still have no idea what they do. And in this particular case, on chromosome 5, there were these 3 genes that people didn't think, well, they were genes, but that was about all they knew. And it was only through the experiments that were done in a model system that led to the idea, that, in combination with human genetics, that led to the idea that this part of the human genome might be dedicated for the formation of this particular cell type. And so I'll stop there and thank Jennifer Stubbs and Lena Ma, 2 postdocs in the lab who have worked on this project, and our collaborator in Germany, Dr. Helmut Amran, who is the clinician who works on primary ciliary dyskinesia. And I'll thank you for your attention and again for your support of the Institute.